Okay, hello and a warm welcome to everyone. Welcome to the first seminar here, the first webinar in our series to highlight the new horizons in circuit neuroscience, both in the field and across the world. So welcome to this very first Inscopix New Horizons webinar series. I'm Kunal Ghosh, CEO and founder of Inscopix. I hope that you're all doing well and staying safe across the world as we continue to socially distance during this pandemic in 2021. Today, we're very excited to launch a new webinar series here, the New Horizons series, led by world-class neuroscientists, our colleagues, and our Asian partners. We hope that this new neuroscience series will provide a sense of community and provide a bridge to our many graduate students and postdocs in Asia who may feel distanced during these difficult times. Before we begin, I just wanted to mention that attendees can submit questions at any time using the Q&A box below. We will be collecting questions throughout the webinar and will answer as many as possible at the end of the presentation. We do have a hard stop at the end of the hour, so if there are questions that we don't get to, please know that it is only due to a lack of time. With that, it is my absolute honor and privilege to welcome our very first New Horizons webinar speaker, Dr. Mark Schnitzer from Stanford University. Mark is a professor of biology and applied physics, an HHMI investigator, and perhaps most relevant and most special to Inscopix and myself, he is also our scientific co-founder. As you all know, Mark was my own personal mentor in the field of neurobiology and neuroscience at Stanford. Over the years, his laboratory has pursued the innovation of novel optical imaging technologies and their use to understand neural circuits. Together, we have created what is now known as the Inscopix Miniscope. In today's webinar, Mark will talk about the motivation for developing the Miniscope, this very crucial tool in the field of circuit neuroscience. And he will also then discuss some of the latest frontiers from his lab in optical brain imaging and some of the new biological results that his own lab has attained in the recent past. The topic of Mark's webinar today is optical studies of neural coding in behaving animals, from miniature microscopes to fluorescence macroscopes. Thank you so much, Mark, for joining us and for kicking off this inaugural webinar series.
Thanks so much, Kunal, for the invitation and for that kind introduction. Welcome to all the attendees. I'm going to share my screen now. And so hopefully you can see the slides. And so as Kunal said, I'm going to give something of a personal view of optical studies of neural coding, and I'm going to discuss miniature microscopes as well as fluorescence macroscopes. And here in this opening slide, you can see large-scale activity visualized with a fluorescence macroscope across one of the transgenic animals produced by the Allen Institute, expressing GCAMP brain-wide. So we'll get to preparations like this a little bit later on in the talk. I'm also going to discuss the miniature microscope quite a bit. Many of you are familiar with this technology, and here you can see an example through a montage of videos in which a set of animals are wearing the head-mounted miniature microscope, and they can naturally behave. Now, one of the things I'm going to focus on today are studies that help us understand how the miniature microscope can reveal facets of neural ensemble coding, as well as dynamical signatures in the brain that are indicative of brain health and disease. And this is going to be one of the major themes that I'll be discussing. So we're going to be looking at these signatures as well as neural coding. So here is an outline of my talk today. First, I'm going to give a brief review of some of the relevant methods that I'll be discussing, in particular microendoscopy and the miniature microscope, in which I and my colleagues have had a hand in developing. Secondly, I'm going to illustrate how the miniature microscope has been a key in helping us to reveal neural ensemble signatures of brain disease. And I'm going to try to illustrate two different types of signatures. One, which I'll call activity imbalances, arises in examples such as Parkinson's disease and dyskinesia, in which there are gross changes in the level of neural activity that are indicative of different disease states and that we think may cause different symptoms, in these two diseases, movement symptoms. And another set of examples is what I'll call altered coding. So there can be aberrant neuronal coding that can give rise to symptoms. And I'll illustrate this using chronic neuropathic pain as an example. During the latter portion of the talk, I'll make some brief remarks about fluorescence macroscopes as a way of visualizing even greater numbers of neurons in more brain areas than can typically be achieved with the miniature microscope. And in the final portion, I'll give an example of a kind of study that one can perform with a fluorescence microscope. In particular, we used a 16 laser beam 2-photon microscope to interrogate thousands of neurons in the primary visual cortex of the mouse. And I'll show you how we found some new evidence indicating the accuracy limits of sensory cortical coding in the mouse. So that's the plan for where we're going. Now here in this first part, I want to briefly review some of the relevant methods. And in particular, I want to point out that in many studies across different systems in the brain and across many different areas of inquiry in neuroscience, there can be a general need to combine different methods for tracking neural ensemble activity. So for example, in many studies, one wants to be able to image large cohorts of neurons, whether or not they lie in superficial or deep areas of the brain. In many cases, we want to be able to track large sets of individual neurons over multiple days and monitor how their patterns may be changing over the course of learning or disease progression. We want to be able to monitor the large-scale activity patterns of these neurons in natural behavioral conditions in freely behaving animals. And something I'll just touch on briefly in the talk, we need to have computational approaches and pipelines that are up to the job of analyzing the large data sets that these imaging methods can produce. And Inscopix, of course, has been very active in bringing these kind of computational analyses to the community. One of the things I'll try to illustrate by way of example in my in the disease studies cases that I'll be discussing later is that by putting all these methods together, there are new possibilities for tracking neural dynamics across time and across different brain states, and in particular normal states and brain disease states. And you'll see that illustrated a little bit later. But right now I'm going to illustrate very quickly and discuss these different methods that we want to combine in a great number of studies. So how do we image neurons that may lie deep in the brain? The challenge here is light scattering. Many people think it's absorption, but actually it's light scattering that is the chief impediment to the penetration of focused light or ballistic light in the brain. And this sagittal view of the rat brain illustrates the limited penetration depth of conventional light microscopy, either confocal microscopy or conventional 2-photon laser scanning microscopy. And you can see that even the 2-photon microscope, which uses infrared illumination, can only penetrate maybe 500-600 microns into the brain tissue in normal experiments. There are ways to extend this, but still one is usually looking at the outer surface of the brain. So a question that we tried to address years ago in our lab was how are we going to image cells that may lie in deeper areas? And the way that we came up with was to use microendoscopes, and in particular fluorescence microendoscopy, which has now become widespread across the neuroscience community. So this slide illustrates some of the original microendoscopes that we designed. They come in all different sizes and diameters. So on the one on the left, it's illustrated here is 1,000 microns across. This one is 350 microns in diameter. And these two endoscopes, you can see an objective at the tip, followed by a longer but weaker relay lens, whose purpose is to give us the reach we need to penetrate into tissue. And these can provide us micron scale resolution that we need to see individual cells deep in the brain. And they can be implanted or inserted directly into brain tissue. So we started this work around 2002, first published 2003, but nowadays neuroscientists use thousands of these probes every year, many of which are made available to the neuroscience community by Inscopix. And I'm going to show you here, just by way of personal history, some of the very first images of neurons that we attained through microendoscopes. This is work done when I was at Bell Laboratories before I got to Stanford. Some very early images here from 2003, and some of our first in vivo images in the rodent hippocampus through microendoscopes. Now, by about 2011 or so, we had begun to routinely image neurons over extended time periods in time-lapse experiments through microendoscopes, either by permanently implanting the lens itself or by implanting an optical guide tube. And here you can see an example of this from the CA1 area of hippocampus. This is a mouse that's expressing a fluorophore in a subset of CA1 pyramidal cells. And you can see from these example time-lapse image data sets that we can come back again and again to individual neurons and individual dendrites over time, and find these same processes with very high precision of return through the microendoscope. And this kind of precision of coming back to the same neural elements repeatedly is very important for many studies, such as the studies of disease that I'll be discussing later on in the talk. Now, a third important development was the creation of the miniature microscope itself, as Kunal mentioned, and this was actually his PhD dissertation, the version shown here, about 2 grams in mass and based entirely on semiconductor optoelectronics. And here you can see one of the early units on the tip of Kunal's finger. Now, interestingly, we had started not with a 1-photon miniature microscope, but even earlier, we had actually started by trying to develop a miniature 2-photon microscope. For example, with Stanford professor Olaf Solgaard, and this is an example of a miniature 2-photon microscope next to a US dime, and we had delivered ultra-short infrared pulses through one pathway in this system, and we're collecting fluorescence light through a different pathway, and we were accomplishing the laser scanning using silicon micromachined microelectromechanical laser scanning mirrors. And there are still groups actually using this kind of miniature 2-photon microscope approach. But at some point, we realized that the CMOS cameras that nearly all of us carry about in our cell phone cameras had become so good that there was the possibility of creating a miniature microscope that was entirely integrated and that contained all the optics and all the optoelectronic parts on the mouse's head in a unit the animal could bear while it was freely behaving. And we realized how powerful this would be rather than having, for example, a standalone benchtop laser like you would have in a 2-photon microscope. So this was work done by Kunal Ghosh and also his graduate student colleagues, Laurie Burns and Eric Cocker, in collaboration with our Stanford colleague, Professor Abbas El-Gamal. This is the miniature microscope that has become the basis for a large part of the Inscopix product line. And here you can see the different components of the system, the CMOS camera here, LED illumination source on the side, miniaturized dichroic and filter sets, a miniature objective peeking out here. And this can be used in conjunction with microendoscopes that are then chosen and targeted to the brain area that is pertinent for the experiment at hand. So this is an example of a mouse wearing the microscope. You can see up close that the animal can behave quite naturally despite the head mount. And meanwhile, we can be streaming large data sets about neural ensemble activity. Okay, and so this is the kind of technology that gives rise to the experiments that I illustrated at the beginning of the talk. And which has revealed patterns of neural activity in a wide variety of different behaviors and a wide variety of different brain areas. And with this technology in hand, Inscopix spun out of Stanford University, and along with myself, Kunal, Laurie Burns, Eric Cocker, and Abbas El-Gamal initiated the company, and subsequently we've been able to serve broad sectors of the neuroscience community. So there have been many applications of this kind of technology throughout different areas of brain science, in sensory neuroscience, in motor neuroscience, in areas of social neurobiology and cognition, combining these different approaches that I've discussed: deep imaging, imaging in freely behaving animals, longitudinal imaging, and of course, a lot of the computational approaches that are so critical to the studies that make use of the ability to image large neural ensembles. Now, in the second part of the talk here, I'm going to illustrate how the miniature microscope and longitudinal imaging deep in the brain have allowed us to examine different signatures of different brain states, and in particular of different brain disease states. So the first category of a brain disease signature that I'm going to discuss is what I've been calling activity rate imbalances. And this is a case where you have a frank or gross imbalance in a proper level of neural activity. And I will illustrate this in a study that looked at the basal ganglion, in particular the dorsal striatum. And this was work that was done in collaboration with Mike Ehlers, as well as Jones Parker, Jesse Marshall, and Biafra Hananu. And what I'm going to show you is that we found opposite types of imbalances in neural activity levels in a Parkinsonian brain state and in a dyskinetic brain state. This was published in 2018. We called it diametric neural ensemble dynamics in these two states. And perhaps another way of describing this is that we found anti-symmetric sets of deficits in these two different brain states in a way that I'll make clearer as we go along. So I think most members of the audience are probably familiar with the idea that dopamine neurons in the substantia nigra pars compacta project to the dorsal striatum. And here in the dorsal striatum, the recipients of dopamine signals are the spiny projection neurons of either the direct or indirect pathway of the basal ganglia. And I'll be calling these the DSPNs and the ISPNs in this talk, although sometimes you'll hear the phrases or the terms D1 MSNs and D2 MSNs, which indicate the two different versions of medium spiny neurons. And the reason for that is that these two neuron classes typically express either the D1 dopamine receptor variant or the D2 dopamine receptor variant in the direct and indirect pathway, respectively. Now, the conventional thinking about how the activity patterns in these two cell classes relate to movement was encapsulated in what's often called the classical model of basal ganglia function. And this was formulated in the mid to late 1980s by people such as Ma Long DeLong and others. And the idea was that activity in the direct pathway, activity of the DSPNs, would serve to activate motion by disinhibiting thalamocortical motor programs, whereas activity in the ISPN population would serve to suppress motion by increasing inhibition of these thalamocortical motor programs. Now, the idea for why the depletion of dopamine in a Parkinsonian state might lead to an impediment of movement was explained by the rate model through the idea that perhaps the loss of dopamine imbalances the rates of DSPN and ISPN activity levels, leading as one logically propagates the consequences of this purported imbalance through the different nuclei of the basal ganglia to increased inhibition of thalamocortical motor programs, as I've said. So that was the hypothesis, namely that you would have hypoactivity in the DSPN population and hyperactivity in the ISPN population. And there had been a number of electrophysiological recordings, many of them in monkeys, that were consistent with this idea. But due to the lack of cell type specific recordings, it had not been directly examined whether or not there actually was the hypothesized imbalance of activity here in the striatum, where the DSPN and ISPN populations of cells are closely interspersed. And so we reasoned that this was potentially an ideal application of calcium imaging using the miniature microscope with genetically encoded calcium indicators, because we could target the indicator to either the DSPN or ISPN populations and directly determine whether or not there was an imbalance in a Parkinsonian state. So we conducted that study using the miniature microscope and tested whether this hypothesis was borne out. And so we did this using different Cre driver lines of mice to target these two different classes of cells before or after depletion of the dopamine neurons in the substantia nigra pars compacta using a chemical lesion. And first I'm going to show you what activity looks like in the DSPNs in a D1 Cre driver line of mouse. And here you can see this is a healthy mouse. And when the animal begins to run or walk, you can see heightened activity here in the DSPN population. And if you look carefully, you can also see that the activity occurs in a spatial temporally clustered manner. You see clusters of cells that tend to activate together. And this correlation between locomotor velocity and activity level, as well as these clustered patterns of activity, is very typical also of ISPNs too. So here I'm going to show you the protocol that we used for our experiment. First, we examined animals while they were in a healthy state. We also tested the effects of various dopaminergic receptor pharmacological manipulations. I'm not going to talk about those today. We then used a lesion to ablate the dopamine neurons in the SNC. We tracked the animals in the Parkinsonian state. We treated them with some of the treatments that are actually used in human patients, in particular dopamine receptor agonists, as well as L-DOPA, a dopamine precursor. And finally, at the end of the experiment, we looked at a dyskinetic state by delivering the animals a very high level of L-DOPA, which causes dyskinetic movements, as in the Parkinson's disease patients in the late stages of their treatment with L-DOPA. So we tracked individual animals through all these states using these chronic preparations that I mentioned in the beginning. Now, in normal mice, we found that there were very similar relationships between activity levels in these two neuron populations and the animal's running speed. We found that at motion onset and offset, the two populations activated and deactivated in a very similar way. And we also found by quantifying a statistical measure of the spatial coordination of the activity in these two populations, that clustering of SPN activity was very similar across the direct and indirect pathway cells. So in a normal state, there appears to be a lot of different factors that are balanced between these two different neural populations. We then took a look at what happened after dopamine depletion, 1 day after and at 14 days after loss of the dopamine neurons. And what you can see here at 1 day after is that now we have a situation in which there is hypoactivity in the DSPNs and hyperactivity in the ISPNs relative to before the lesion. 2 weeks after the loss of the dopamine cells, we continue to see deficits in activity in the DSPN population. The ISPN population has partly normalized when the animal is moving, but there remains a substantial elevation of activity in the ISPNs, even when the animals are at rest. Now, interestingly, the activity levels are not only abnormal in the ISPN population, but they have also lost their coupling to movement. So in the DSPN population, despite the fact that they are hypoactive, they still respond when the animal accelerates, for example, whereas we see at 2 weeks after dopamine depletion, this is not the case in the ISPN population. So I'm looking at this pink curve here. So we also saw a similar deficit at motion onset and offset. Again, the ISPNs seem to fail to respond to changes in locomotor velocity, signifying that they are uncoupled from movement. And again, we also found that their spatial coordination no longer changes in a normal manner. Now, I'm not going to go through all the details of this chart, but the summary is that we then tried to treat the altered movement patterns in these animals, as well as the altered patterns of neural activity. And we found that the best treatments that work well in the clinic, namely L-DOPA and D2 receptor agonists, are those that work well in our mice. And in particular, L-DOPA was the best at restoring normal patterns of neural activity, even more so than D2 receptor agonism. Now, what I'm going to show you is that when we deliver a high dose of L-DOPA, the animals become dyskinetic and they exhibit involuntary movements of the face, of the trunk, of the limbs that one can score in a manner that's established in the field. And so these involuntary movements are modeling those seen in the late stages of Parkinson's disease treatment with L-DOPA. Now, interestingly, in this brain state, we see an opposite state of affairs than that observed in the Parkinsonian state. So in particular, now we see a hyperactivity in the direct pathway cells and hypoactivity in the indirect pathway. So this is the opposite set of neural abnormalities. And it is very interesting because this brain state dyskinesia is characterized by an opposite set of movement symptoms. Now we have involuntary hyperkinetic movements, whereas in a Parkinsonian state, this is characterized by impediment of movement. And this montage of videos summarizes what we found across these three different brain states. So here you have at the top row a D1 Cree driver line of animal that was taken through these three different states. This is a Cree driver line that is labeled the ISPNs. And here, when the animals are in the normal brain state, you can see that activity levels are approximately balanced and they both exhibit these spatial temporal clustered patterns. Whereas in the Parkinsonian state, here you can see that there is diminished activity in the direct pathway and hyperactivity here in the indirect pathway. And some loss of the clustering is evident here. Whereas in the dyskinetic brain state, we have the opposite imbalance. We have hypoactivity here, hyperactivity here in the direct pathway, and the loss of the clustering. So a fully anti-symmetric set of deficits between these two different disease states, illustrating this imbalanced activity state of affairs that I mentioned. Okay, so that's one example whereby we can see brain signatures of disease. And now I'm going to give you a second example that's a little different. So here in this case, we're going to be looking at abnormal neural ensemble coding. And so our work in this started in a study of the basolateral amygdala, a collaboration with Andreas Luthi's lab in Basel, led by, at that time, postdoc Benjamin Greve. He's got his own lab now in Zurich. And we started off with a study of fear conditioning. So we looked at neural ensemble signatures in classical fear conditioning and examined whether or not we could find a signature of a long-term associative fear memory. And in this particular study, we used the miniature microscope, as I've been discussing, to track ensembles of amygdala neurons in this case to see whether we could discern the representation of an associative memory in this classical fear conditioning model that should be very familiar to many of you. So we're going to be pairing a initially neutral tone known as the conditioned stimulus with an inherently aversive foot shock, the unconditioned stimulus. And this is this slide here shows you our behavioral protocol. It's very standard. We have 2 days of habituation to the fear testing chamber. Third day is when we do the pairing between the auditory tones, the CS, and the foot shock. And then we have 3 subsequent days of fear testing. Now, to cut right to the chase, what I want to show you is that throughout the entire experiment, we were examining the animals during both fear learning and some portions of fear extinction. We found that the extent to which the representation of the initially neutral tone that was paired with a shock, which we called the CS plugs, the extent to which the representation of this stimulus mimics that of the foot shock that we gave the animal was predictive of the extent to which each animal exhibited learned freezing behavior, which is the characteristic conditioned response in this assay. So if you look at this data, each data point here represents the responses from an individual mouse. And what we found was that during both learning and extinction, the extent to which the representation of the CS resembled that of the US was predictive of each animal's level of conditioned freezing. And we thought that was exceedingly interesting because it shows us that even after fear conditioning, the encoding of this initially neutral cue, this auditory CS, is now, after training, resembling the representation of an aversive stimulus, even in the absence of the latter. So now, after training, there is no foot shock, there is no unconditioned stimulus, and yet when we play tone to the animal, the amygdala is giving us a pattern of activity that looks like that of that representing the shock. So it's a wholesale transformation of the representation of this initially neutral stimulus. And so we wondered whether or not a similar phenomenon might occur in the basolateral amygdala in a chronic pain state, and perhaps there might be a similar recoding of benign or innocuous stimuli that it might account, we thought, for the clinically known phenomenon of allodynia. And this is something that chronic pain patients report, in which stimuli that most of us would describe as innocuous or benign are perceived by chronic pain patients as painful. And we wondered or not whether there might be a similar recoding, such as that seen in the fear conditioning experiment. So with colleagues Greg Scher, Greg Corder, and Biafra Hanano, we took a look at this hypothesis, again, in the basolateral amygdala, and we found an ensemble of cells here that encodes the affective or the emotional components of pain experience, these unpleasant components that make pain so aversive for us. So we went into the study first trying to characterize the responses of different BLA neurons, and we found that there were different cells that responded to appetitive stimuli, aversive stimuli, and noxious stimuli. We tracked neural responses in this area, again using the microendoscope and the miniature microscope, technologies that we've been discussing throughout the talk. Did this in behaving mice. And here you can see some example responses of neurons in the basolateral amygdala to noxious stimuli of different kinds: noxious heat, noxious cold, noxious pinprick, and one example with light touch. And what we found was that there was a subset of cells that were selective for noxious stimuli, but not for other aversive stimuli. And here you can see, using a decoding approach, that there was a general similarity in how different types of noxious stimuli were encoded, but this is separate for how other types of aversive but not noxious stimuli were represented in the amygdala. So there seems to be at least a subset of cells that are dedicated, it seems, to encoding nociceptive stimuli. And moreover, we found that the activity levels in these nociceptive neurons in the amygdala were indicative of the animal's level of pain behaviors, paw withdrawal, escape behaviors, and so forth. And so it seemed like the activity in these neurons could account for pain behaviors. And moreover, we showed, using a combination of genetic trapping and chemogenetic silencing, that by inhibiting these neurons, we could achieve a selective reduction in the affective or motivational components of pain behavior, but not the reflexive types of pain behavior. So I'm not going to get into all the details of how we targeted and silenced these cells, but what you can see here on the left is that reflexive behaviors, i.e. paw withdrawal, for example, to a pinprick, were not reduced after chemogenetic inhibition of this cell set, but affective behaviors such as nursing a paw or trying to escape the testing chamber altogether were reduced after chemogenetic silencing of this set of neurons, indicating that they have a causal role in this sort of called active affective set of pain behaviors. And so we were very interested in what might happen to the coding properties of these neurons in a chronic neuropathic pain state. And so we examined the mice using a spared nerve injury model of neuropathic pain. So there's a peripheral nerve injury, and we were particularly interested in the phenomenon of allodynia, which, as I said, is when normally benign stimuli are perceived as painful. And so we imaged mice before and after a nerve injury. And first, I'm going to show you using behavioral data that in the chronic neuropathic pain state, animals exhibited behavioral signs of allodynia. So when we applied light touch, reflexive withdrawal behaviors were greatly increased in the neuropathic pain state relative to the normal condition. And when we used the miniature microscope to image neural responses in the basolateral amygdala, we found that the representations of benign and nociceptive stimuli were much more similar in the neuropathic state, a little bit akin to what I had showed you before with the CS and US representations in the fear conditioning experiment. And what we found, for example, is that the responses of neurons that normally would be responsive to light touch, now we saw that light touch after the nerve injury would trigger activity in the nociceptive population. So nociceptive neurons in the BLA were now responding to benign stimuli in a way that seemed abnormal. Now, to show you that these neurons and these abnormal responses had a causal role in chronic pain-related behaviors, we then again trapped and chemogenetically silenced neurons that were responsive to light touch. And this had no effect on animals' reflexive pain behaviors, i.e. paw withdrawal, but silencing of this light touch responsive set of cells substantially diminished the affective motivational set of pain behaviors to light touch responsive stimuli. And this effect of silencing these neurons in animals that were in a chronic neuropathic pain state, I think is perhaps most vividly illustrated with these data here, where we placed the animals on a cold plate assay. So on the left, you can see that there's a plate at 15 degrees Celsius, on the right, control plate at 30 degrees. And the chronic pain animals will avoid the left-hand side, but when we chemogenetically silence this particular set of cells in the BLA, now the animal will happily explore both sides of the plate, indicating that it is no longer experiencing the cold side as painful. So we have now identified, and through this chemogenetic approach, at least in mice, it seems like we can really address the animal's chronic pain experience, and in this particular case, the allodynia. So to sum up this part of the talk, I've tried to illustrate two different types of neural signatures of brain disease that the miniature microscope can reveal. In the first category, we have activity imbalances, and I showed you that across the direct and indirect pathways of the basal ganglia, there were two different types of imbalances in a Parkinsonian and a dyskinetic state, and that these imbalances could be bidirectional and spatiotemporal in their character. So we also had disruption of the spatial-temporal clustering patterns of activity. Treatments such as L-DOPA can rebalance activity and restore normal patterns of behavior and restore normal neural activity signatures. And I think what was perhaps one of the most interesting facets of this is that we looked at two different brain states which are characterized by opposite sets of movement symptoms, and they seem to be characterized by opposite imbalances. So here, in this type of situation, it seems that the therapeutic goal is to try to rebalance activity. By comparison, in the second example that I showed you in the basolateral amygdala, we seem to have abnormal coding. So in this case, unlike the first, the gross or macroscopic levels of neural activity seem to be unchanged. Rather, it's that the patterns of neural coding and their causative role in creating behavioral and affective experience are altered. And so here it seems that the successful treatment approach is to try to target the specific cell class that may exhibit abnormal coding properties and to try to renormalize that cell class, as we did using the chemogenetic approach in the chronic pain studies. So this, I think, gives you some examples of how our newfound ability in recent years to visualize neural ensemble patterns of activity in freely behaving animals can give us real insights into different abnormalities in different forms of brain disease. Now, as we head into the latter portion of the talk, I'm going to discuss some new technologies and new approaches that can help us visualize very large-scale neural ensemble dynamics, larger even than the typical miniature microscope experiment can provide, either in terms of numbers of cells or in terms of numbers of brain areas. Why do we want to do this? Well, a lot of work in the neuroscience literature has looked at different levels of organization in the brain, at the synaptic level, cellular level, at schematic brain circuit models. And what I've been showing you up to this point in the talk was that sort of neural ensembles, microcircuits within a given brain area. But of course, we'd like to understand how neurons in different brain areas may work together in concert. We'd like to understand multi-area networks of cells. So it's going to be very important to develop imaging techniques that can take us to this level of inquiry. And I'm going to show you a few examples of how that can be done. So one very simple means for probing more than one brain area is to make use of the small size of some of these miniature optics to interrogate more than one brain area at a time. And this can be done, for example, by implanting more than one microendoscope or by using them within a multi-arm two-photon microscope. So this is an example that was built by Jerome Lecoq when he was in my lab. This is a double-armed two-photon microscope. Each arm can be flexibly moved, each arm terminated in micro-optics so they can be positioned to look at either nearby or distal sets of brain areas. And I'm going to show you what this looks like in practice. So this is data from a collaboration with my Stanford colleague, Li-Chun Lo, and led by Mark Wagner and Tony Kim in the lab. And here you can see a head-fixed mouse under this double-armed two-photon microscope. The animal's pulling a lever here, a robotic manipulandum. And meanwhile, we are imaging neural activity by using calcium indicators in the cerebellar granule cells, and then another population in the motor cortex, the layer 5 pyramidal neurons. And so we looked in this study how these two different sets of neurons began to coordinate their activity patterns over the course of motor learning. Now, another approach for looking at more than one brain area at a time comes through this chronic mouse preparation that we call the crystal skull. And in this approach, Tony Kim and Yanping Zhang showed that they could permanently replace a large portion of the dorsal cranium of the mouse with a curved glass window, whose radius of curvature was matched to the natural curvature of the brain. So I'm going to show you what this looks like. This is a transgenic mouse expressing GCAMP in layer 2-3 cortical pyramidal cells. The animal is running, and we're looking across the cortex with a fluorescence macroscope. And those little twinkling spots you're seeing are somatic calcium activation of individual layer 2-3 pyramidal neurons. We can zoom in, and here you can see, and with this magnified view, you can see activity in the individual neurons more closely, as well as some of the blood vessel dynamics, such as the constrictions that occur when the animal speeds up. And we can zoom in yet again. So here, for example, on the right panel, you can see a magnified view of what's happening here in this inset. And now at this magnification, you can very clearly see the activation of individual pyramidal cells in this so-called crystal skull preparation. And so this is a very valuable tool for gaining optical access to many neurons in many different brain areas. And in fact, we've shown that this preparation can allow you to sample cells in between, say, 35 to 40 brain areas at once. This is using the Allen brain map as a reference. Okay, now another approach that we have developed to look at large numbers of cells is this particular two-photon microscope built by Oleg Rumyantsev, Jerome Lecoq, and Joan Saval. It has 16 laser beams, which are multiplexed in space and time from a single ultra-short pulse titanium sapphire laser. And They tile the field of view in a 4x4 checkerboard pattern. So we have 16 image tiles and 16 corresponding photomultiplier tubes for photo detection. And the reason we have this simultaneous interrogation of these 16 portions of the image is it allows us to scan a greater field of view per unit time than we could do with a single laser beam. So I'm going to show you what this looks like. With this system, we have a field of view of 4 millimeters squared. We can interrogate it at frame rates of about 20 hertz. And this is enough to encompass an entire primary visual cortex in an awake mouse, allowing us to probe the representation of an entire visual scene. And here you can see what the data look like from a transgenic animal made at the Allen Institute by our collaborator, Honghui Zhang, targeting the GCAMP indicator to layer 2-3 pyramids. So I'm going to show you a movie of what this looks like using the 16 beam system. And if you look carefully, you might be able to see the 4x4 pattern of image tiles in this movie. You can see the somatic patterns of calcium activation, and those dark stripes are the blood vessels that appear in relief. I'm going to show you another movie. This is a case in which you can plainly see the dendritic activity as well. And so these fingertip-like excitations are patterns of dendritic activation that we can see. So with this system, we then had an opportunity to examine a question that could only be probed if you had recordings of 1,000 or 2,000 neurons per animal. And that's a question that had been around for a long time in the neuroscience literature. What limits the accuracy with which a large population of neurons can encode a sensory scene? And we published this recently. It's a study of primary visual cortex V1 in the mouse. And I'll show you why this study, why addressing this question required us to record from thousands of neurons per animal. So it had long been asked, what are the factors that bound the fidelity of sensory neural coding? And there had been a long-standing set of ideas going back to actually my Stanford colleague, Bill Newsom and his coworkers, that perhaps correlated noise in 2 or more cortical neurons might actually set an upper limit or a bound on the fidelity of sensory cortical coding. And so what I'm going to show you is that we were able to obtain direct evidence for such an effect in ensembles of 1,000 or more cells, that the levels of correlated fluctuations that we measured in the mouse is consistent with the animal's visual acuity, and that the pattern of noise gave us some insights into how the cortex computes accurately despite substantial noise fluctuations. Now, work in the peripheral visual system, such as in the retina, had suggested that for weak sensory stimuli, the limits on representing a scene would be set by the primary sensory neurons, the photoreceptors, the retinal ganglion cells, and their sensitivity to individual photons, essentially. But for stimuli with greater numbers of photons, Newsom and Mike Shadlin, Tony Mofshin, and their colleagues hypothesized in the late 1980s and early 1990s that perhaps the limit on the brain's ability to represent a sensory scene might be set by correlated fluctuations amongst cortical neurons. And they did some seminal calculations. So here on the x-axis, you can see a plot which is a function of the number of neurons in an ensemble that you might consider, and the signal-to-noise ratio that you can attain from such an ensemble under different conditions with no correlations in the fluctuations of the cells or at increasingly greater levels of fluctuations. And what you can see is that even for small amounts of correlated noise, correlated fluctuations, there is ultimately a plateau or a bound on the signal-to-noise ratio that is achieved. And you'll also notice that you may need very large numbers of neurons in your recording to be able to see this effect directly. Now, in these original studies, Newsom and colleagues had assumed that the neurons would have identical sensory response properties, but the analysis generalizes nicely to ensembles with more diverse tuning properties. And I'll just illustrate that quickly here for the world's simplest ensemble of 2 neurons. So imagine we have 2 cells, and we show the animals one of 2 different stimuli, and we record the joint responses of these 2 neurons. So we have an orange distribution of data points and a blue distribution indicating the joint responses of these 2 neurons to repeated presentations of the 2 stimuli. We can then ask, how well can we discriminate these 2 stimuli based on the 2 cells' joint responses? And whether we have uncorrelated noise or positively correlated noise or negatively correlated noise, the component of the noise that is important is that which lies along the tuning curve, which is indicated in these diagrams by this red dotted line. And so this is true in all three cases. And generally, one wants to draw a decision boundary between these two distributions. So you play a stimulus again, and then depending on whether the joint response lies on this or this side of the decision boundary will allow you to determine which stimulus was played. And it's the noise component perpendicular to the decision boundary that's going to determine the fidelity with which you can make that discrimination. So what had been missing in this field was a direct test of whether or not this upper limit on signal to noise ratio was actually a bona fide effect in large cell ensembles. And so we set out to test this in mouse V1 using our large microscope. And so this was a study that we completed recently. It was a collaboration with my Stanford colleague, Surya Ganguly. And the way that it worked was as follows. We had mice sit under the microscope while we presented them with one of two different stimuli on a video monitor, moving gratings that were oriented at either plus or minus 30 degrees relative to vertical. Each stimulus was shown for 2 seconds, and the stimuli were ordered in a random pattern, random order in time. And meanwhile, we recorded the pattern of evoked responses in V1 layer 2-3 pyramids. We then asked the question from the thousands of neurons recorded in 6 mice, how well we could distinguish the two sets of stimuli from the neural ensemble activity patterns. Now, one of the challenges in this study was the fact that we have to be able to determine accurately the correlated noise patterns of this large number of cells. So imagine that we have in each animal 1,000 to 2,000 recorded cells. That is going to lead to a noise covariance matrix which has a size on the order of a million elements or more. So we're not going to have enough data for that if we have only, say, 200 to 300 trials per mouse, because that's only going to give us on order of magnitude p times n measurements. So we can't determine the elements of the noise covariance matrix directly. On the other hand, perhaps we can take a decoding approach. To specify a stimulus decoder, at least a linear one, only requires n parameters. So with that perspective, perhaps p times n measurements may be sufficient. And so again, I'm going to illustrate this point with the world's simplest ensemble. So imagine we have 2 cells in our ensemble, and we can record their joint responses to 2 different stimuli, as indicated here by the 2 different clouds of data points. And we can then build a linear decoder by drawing this hyperplane. So with n cells, we would do this in an n-dimensional space. We draw this decision boundary. And the fidelity with which we can make this discrimination is determined by delta mu, the distance between the centroids of these 2 distributions, and sigma, the noise amplitude along the neural ensemble tuning curve, i.e. perpendicular to the decision boundary. So in practice, we did this using a partial least squares analysis, which allowed us to find neural decoders that accounted for the patterns of correlated noise in a space of reduced dimensionality. And I'm going to show you what this looks like with real data. So here I'm just showing you 2 different dimensions from the partial least squares analysis. At 0.15 seconds after we showed the mouse a visual stimulus, and at 1 second. And here you can see very early on, after the stimulus is shown, that the distributions of neural responses to the 2 stimuli cannot be well discriminated, but a little bit later they can. And this green line here is the optimal decision boundary that takes into account the patterns of correlated noise. You can see the correlated noise very clearly. These noise distributions are asymmetric and at an angle, whereas here is a decision boundary or a decoder that does not take into account the correlated noise. So the key question was then, how does D prime, our fidelity of decoding, scale with n, the number of neurons in the ensemble? And specifically, does D prime plateau, as was predicted by Newsom and colleagues? Now, we have a very important control when we do this kind of analysis, which is that if we shuffle the data, if we create trial shuffle data sets, which do not have correlated fluctuations amongst cell pairs, then we should see no plateau at all in D prime with large numbers of cells. So now I'm going to show you the results. So here on the x-axis of these plots, we have the number of neurons, and we use 2 different decoding approaches. An instantaneous approach in which we looked at neural ensemble activation at each time bin after a visual stimulus onset, and a cumulative decoder in which we looked at all the activity up to a given point after a stimulus onset. The results were the same. So we found in the trial shuffle data sets that there was no evidence of a plateau. Each curve here is from an individual mouse, whereas in the real data sets, we found a plateau in every mouse. And you can see that after we have ensembles larger than 1,000 neurons or so, there's a clear saturation in each and every animal. So it seems as if that you need about 1,000 neurons or so to optimally decode, but after that, more neurons are not helpful. We repeated the experiment with 2 different stimuli that were a little harder to discriminate. Gradings oriented at plus or minus 6 degrees. We again saw the same results, a plateau in the real data, but not the shuffle data. And these data sets allowed us to estimate the minimum resolvable orientation difference that a mouse might be able to discern. This estimate was about plus or minus 2.4 degrees, which was actually very close to the prediction from psychophysical visual acuity measurements in the mouse. So it seems as if the D prime measurements that we're taking with our microscope are consistent with numbers from mouse visual behavior. Finally, what causes the saturation at large numbers of neurons? And in particular, are there eigenvectors of the noise covariance matrix whose eigenvalues grow linearly with n, just like the visual signals, that will cause the plateau in SNR? And so what this slide shows you that the answer is yes. Here you can see the growth of visual signals as the ensemble gets bigger. And here in the real data shown in the green, each mouse also has an eigenvector whose eigenvalue, a noise eigenvector whose eigenvalue is also growing as the ensemble gets larger. The ratio of these two is what gives us the plateau. This does not occur in the shuffled data. Now, what was interesting is that not only did each mouse have this pattern of eigenvalues of the noise covariance matrix that grow with the ensemble, but it was not actually the largest noise eigenvalue. In fact, typically it was the 3rd largest noise mode. So you have to think about the structure of noise in the high dimensional space at which these neurons are representing the visual stimuli. So we have the tuning curve, and as I showed you earlier, that was the direction that's important. But in fact, it was the largest noise mode. This largest noise mode was orthogonal to this critical direction in space, the tuning curve. And it was the 3rd largest noise eigenmode that was aligned to the visual signals. So interestingly, when we quantified this effect, 90% of the noise was orthogonal to the visual signals and thus did not impede coding. It was the residual 10% of noise that was that set the bound on the accuracy of the visual code. So to sum up what we found direct evidence for the predicted plateau in populations of 1,000 more cells, confirming the classic predictions by Shadlin, Newson, and others. Every mouse had a few noise modes that grew linearly with the size of the neural ensemble, as did the visual signals. It seems that correlated noise is what's actually limiting the accuracy with which a sensory scene can be represented. These noise patterns were consistent with previous measurements of mouse visual acuity. And interestingly, 90% of the noise that we saw in the visual cortex was actually orthogonal to the visual signals, suggesting that rather than removing noise altogether, the brain may be improving visual coding accuracy by doing something very clever. It's putting the majority of the noise into dimensions that don't matter and therefore don't interfere with the accuracy of sensory encoding. So this experiment, I hope, shows you an example of one application of a macroscope in which you really need to be able to analyze not just hundreds, but 1,000 or more neurons at a time. So to sum up the entire talk, I've shown you quite a lot. I've shown you that miniature microscopes can access patterns of neural ensemble activity. And this is true in a wide variety of brain areas in free behaving animals. I gave you 2 different examples of how the miniature microscope technology can not only allow us to study neural coding, but also signatures of different brain disease states. In a Parkinsonian and dyskinetic state, we have imbalances, or in a chronic neuropathic pain state, we have abnormal neural coding. I discussed macroscopes as an emerging way to track thousands of neurons, and I showed you an example of a fluorescence macroscope, which has allowed us to look at the accuracy of sensory encoding and pinpoint the fact that it is in fact correlated noise in the cortex that limits that accuracy. And with that, I'll stop, and hopefully we have some time for questions.
Hi, Mark. Thank you so much. This was fascinating. We definitely have some questions. I would like to remind everyone that you are welcome to provide questions to us via the Q&A box. And as people gear up to enter in some questions into the Q&A box, I'm going to go ahead and start with a question here. Recently, there were some publications showing that there was an application of a miniaturized 2P. Since your group also tried that, I'm curious what your perspective is about it. Can you think of some pros and cons compared to the one-piece system that was originally innovated by your group?
Well, I think one of the important ones is the lack of scalability. Hold on, there's something wrong with the Zoom here.
Yeah, I think there you go.
Yeah. So one of the chief advantages of the one photon approach is the scalability. The fact that it's very easy, as I showed you, for example, in that montage of videos from the basal ganglia, to have multiple animals running at once. Whereas with a two photon system, one has to have accompanying benchtop instrumentation that is much larger, requires an Airtable, is much more expensive and so forth, and simply cannot be scaled in the same way. There are other important points. So for example, with the one photon system, you can have large fields of view. We're doing volumetric imaging, and I think typically more neurons can be examined. And concerns about motion artifact are far diminished due to the fact that there's no laser scanning occurring on the animal's head that would be subject, for example, to the torques that would occur as an animal moves about. And that can be an issue in some of the two photon experiments. And also the optical sectioning with two photon microscopy can be a disadvantage in this context. Because it means that neurons can move up or down below the focal plane, and you can momentarily lose them in a way that does not occur with the one photon. So I do think that the head-mounted miniature two photon is an interesting device that is going to have some applications. For example, if you really wanted to look at dendritic spines or dendrites in freely behaving animals, but for many, many experiments, we have found through pragmatic experience that the one photon microscope is more than enough in terms of what you need to look at neural ensemble dynamics, is scalable, so it allows you to have multiple devices, is compatible with optogenetic manipulations, as Scopix has shown and commercialized, and is really can be superior in many cases for the studies that are desired.
Yeah, that's a really good points. Another question we have here is an interesting one. What would be the most challenging question in the in vivo imaging field from your perspective? What kind of milestones would you envision for the neuroscience community that you think could be achievable in the next 10 years?
Well, it's always hard to predict the future, but I think, there are multiple advances in technology, obviously, that we would like to have. So, one obvious set of challenges for those of us that design and build new microscopes is the ability to keep going in the direction that I was illustrating in the latter portion of the talk. Namely, we would like to be able to visualize, still yet more brain areas, different types of neurons. So can we go beyond, for example, red and green channels to do, many different types of neurons? Can we do sophisticated optical manipulations or other manipulations in the system? And are there other facets of neural activity that new indicators might reveal? So I didn't talk much about indicators today, but we've also worked on voltage indicators. This has been a very exciting area of recent progress for the field. I think that there's likely to be upcoming progress in the ability to image not only neuromodulators, for which there are new indicators available, but also neuropeptides. And so, for the protein engineers and chemical engineers who are working on indicators, these I think are going to be important milestones to help us reveal facets of brain activity that we simply cannot see today.
Yeah. I have another question. Do you think has this technology been extended to outside of the brain, for example, enteric ganglia?
Yeah, that's a very interesting question. We have we've received inquiries of that type repeatedly over the years. the cranium or the skull provides a big advantage. It allows one to anchor the microscope in a firm way as the animal is freely behaving. And that's, that's not so easy, for example, in a study of the enteric nervous system. And so that's been an impediment. It's also not clear that you would necessarily want to look at a freely behaving animal if you're studying the enteric nervous system. Perhaps you might do that with a micro lens, but you wouldn't necessarily need to couple that to a miniature microscope. So there have been cases in which people have used these microendoscopes and used these miniature microscopes to look at different parts of the body, including the peripheral nervous system. But it, at least to this point, it hasn't really caught on in the same way that now you have hundreds of groups using the miniature microscope to study the brain.
Yeah.
Perhaps it will in the future, but to this point, it's been more isolated applications.
I think we have time for maybe one more question here. I realize this has been fascinating for a ton of people, and I've got questions galore. You mentioned microscopic activity and macroscopic activity in the neural signatures of disease. Can you comment on how these may interact with each other? Can one address or manipulate one without the other?
Yeah, that's a very interesting question. So, and the question pertains to the elements of causality in the brain, right? So if there's a brain disease and we have symptoms, what is it that actually causes those symptoms? And so although it's common to think about, for example, targets, molecules that may be awry in some manner in a brain disease state, ultimately it is an abnormal pattern of activity of some kind at a macroscopic level that is going to lead to patient symptoms that a patient will actually go to their doctor's office to report. And so at some level, that is the level of organization in the nervous system that we need to understand and to correct if we're going to address symptoms. And so one of the interesting things about the miniature microscope and other ways of visualizing large-scale neural activity is that in the sense that I've been describing, it's a little closer to what actually causes symptoms. So, there may be many reasons, for example, that you would end up with imbalanced levels of activity in the basal ganglia. But by looking at hundreds of neurons, you can discern this imbalance and try to correct it, and thereby it seems correct symptoms.
Right.
So I don't know if that's a full answer, but that gives you an indication of how I think about it.
Excellent. Thank you so much. Folks, I want to thank you all for your participation today in today's webinar. We hope you found this webinar insightful and inspiring. Stay tuned for future webinars. Please feel free to reach out to us on our contact page at www.inscopix.com. I hope you all have a wonderful rest of your day. I'm going to go ahead and close this webinar for everyone. Thanks again, everybody. I'm going to go ahead and end it. Thank you, Mark, for your time and your effort. This was a fantastic day. Appreciate it all. Thank you.